Dear students, our lecture for today is about antigen-antibody interactions. Here are some key definitions will be mentioned during the lecture. Learning objectives, introduction about antigen and antibody interactions. We will explain the terms affinity and avidity, primary interactions between antigen and antibody, secondary interactions between antigen and antibody, and we will mention some types of immunoassays. Introduction about antigen and antibody interactions. Antigen-antibody interaction occurs by combining antigen with antibody specifically. It forms the basis for humoral immunity, antibody-mediated immunity. These interactions are used for detection of infectious agents and some non-specific antigen-like enzymes. Now, what is the affinity and what is the avidity? Affinity, the strength of an interaction between a single binding site on an antibody and its target epitope. Avidity, the measure of the total binding strength of an antibody at every binding site. It also known as functional affinity. Note that IgM have higher avidity than IgG, although the binding of each Fab in IgM with antigen determinant may be the same affinity as that of the Fab from IgG. We mentioned that because IgM is pentameric, that means have more binding sites than IgG for the antigen. Primary interactions between antigen and antibody. It's very important to remember that no covalent bonds involved in the binding. The binding through forces are relatively weak, consist mainly of van der Waals forces, hydrophobic forces, electrostatic forces. Antigen and antibody interaction requires a very close fit. That's very important issue in that case. Very close fit between an epitope and the antibody like a lock and key. Antigen-antibody complex is readily dissociated by low or high pH, high salt concentrations. Now we mentioned some terms, van der Waals forces. It's a weak electric forces that attract neutral molecules to one another. Hydrophobic forces. Nonpolar molecules that repel from water do not dissolve in water and prefer other neutral molecules and nonpolar solvents because water molecules are polar, as you know. Now, reactions between antibody or antibody fragments, antigen or haptens. We have many mechanisms in reaction in that case. Here in this diagram, we have univalent, unideterminant antigen. It's a hapten here in that case. What do we mean by univalent? Univalent antigen have one binding site with the antibody because it have only single epitope. Unideterminant antigen have one single type antigenic determinant epitope. For example, antigen have only epitope A here in this diagram. When univalent, unideterminant antigen, as we mentioned, it's hapten, bind with antibody. Here in this diagram, it's the epitope A, we call this epitope A, and there is antibody specific to that epitope, anti-A, will bind with this epitope, but because of this is univalent, unideterminant hapten antigen, there will be binding with A epitope antigen with anti-A, and complexes will form but not cross-link between each other. There is no cross-linking in that case. Here the antigen is multivalent. Multivalent, that means have more than one binding site with antibody. Either the antigen have many epitopes, but one type of epitopes, just like here. Many A epitopes here on the antigen, 3 epitopes from the same type, because it's unideterminant. Have one type of epitopes, but many epitopes, multivalent. When these multivalent unideterminant antigen bind with anti-A antibody, then anti-A complexes occur and cross-linked between each other. Because here the anti-A have 2 binding sites and the antigen have 3 binding sites in that case, so cross-linked occur in this situation. In this diagram here, multivalent antigen unideterminant will bind with anti-A, but it's defect by act of proteolytic enzymes or something that cut this antibody molecules into some fragments, like single Fab. Anti-A Fab will, when binding with antigen, multivalent unideterminant antigen, in that case anti-A, A Fab complexes occur but not cross-linked between these complexes because there's no other binding site of the Fab to interact with other epitope. Here we have the multivalent unideterminant antigen bind with anti-A, but with 2 Fab bind with each other, as we mentioned, as a result of proteolytic enzymes, papain, pepsin, proteolytic enzyme cut these antibodies into fragment into anti-A Fab2. Two Fab bind together, bind with multivalent unidetermined antigen. These lead to anti-A Fab2 complexes and cross-linked between each other because there's two binding site of these two Fab and the antigen is multivalent. In that case, cross-links occur. Here, the antigen is multivalent, multideterminant. Multideterminant, that means, as we see from this diagram, it's A epitope, we have B epitope and C epitope. Three types of epitopes in that case. When bind with anti-A, we have three types also: anti-A antibody, anti-B antibody, and anti-C antibody. Anti-A is directed to A epitope, anti-B to B epitope, anti-C to C epitope. When these bindings occur between these epitopes and these anti-A and B and C antibodies, anti-A, anti-A, B anti-B, C anti-C complexes occur and cross-links between each other. Secondary interactions between antigen and antibody, lattice formation. Different outcomes may result from secondary antigen-antibody interactions. Agglutination antigen is particulate, precipitation antigen is soluble, activation of complement. Agglutination means the process of clumping of antigen particles by their respective antibodies. We have direct agglutination, in which antigen is natural constituent of a particle, while we have passive agglutination when the agglutination reaction takes place between antibodies and soluble antigen that had been attached to insoluble particle like latex. The precipitation is a process where soluble antigens bind with their specific antibodies at an optimum temperature and pH, resulting in the formation of an insoluble precipitate. Lattice formation means cross-linkage between antibody and antigen, cross-linkage formation between these two compounds. All these outcomes caused by interactions between multivalent antigens and antibodies that have at least two combining sites per molecule. Here are some kinds of examples of agglutination reactions, semi-quantitative. The agglutination reactions characterized by these features, these agglutination reactions are semi-quantitative, performed by mixing two-fold serial dilutions of serum antibody with a fixed concentration of antigen. Agglutination may not occur when antibody presence in excess. We call Prosome phenomena. When antibody present in excess, Agglutination also may not occur when antigen present in excess. Post-zone phenomena. We call post-zone phenomena when antigen present in excess. Now the question here arises. Antiserum tested at several dilution. Why? Think about that question. Okay? Now, zeta potential. The zeta potential occur when the surfaces of certain particulate antigen may possess an electrical charge. For example, the net negative charge on the surface of RBCs caused by the presence of sialic acid. When such charged particles suspended in saline solution, an electrical potential created between particles known as zeta potential. This potential prevents them from getting close to each other. The zeta potential makes it difficult to agglutinate charged particles by antibody. Now, here another questions regarding to zeta potential, which is more effective as agglutinating antibodies, IgG or IgM? Also think about this question. Keep this question in your mind. In 1950s, Combs created a method to overcome zeta potential by employing antibodies to immunoglobulin and anti-immunoglobulin. Here in this diagram, the RBCs as antigen and antibody. When bound together, there is no agglutination because of the zeta potential make these particles far away from each other. By adding anti-immunoglobulin, then agglutination occurs in that case. Example of agglutination reactions test, we have direct Combs, hemolytic disease of the newborn, the HDN, we mentioned that disease in hypersensitivity reaction lecture. Indirect Combs, detection of anti-RH, IgG antibodies in the blood of an Rh negative woman. Also, we mentioned these details in hypersensitivity reaction lecture. And blood grouping pregnancy test. Precipitation and a precipitation reaction in liquid. For example, Widal test for testing infection with Salmonella typhi. Here in this diagram, you see that adding a constant amount of antibody in each tube, then adding increased amount of antigen in each tube. That means we increase the quantity of and concentration of antigen in each tube. Antigen antibody interact and precipitate in the bottom of the tube. Here, first 4 tubes, the first tube there is no precipitate because antibody access zone. Here in these tubes, the precipitate is very the quantity is very small. In equivalent zone, as we note here in this tube, surrounded by red circle, the equivalence zone, that's mean in that tube. Here, the antibody equal to antigen concentration result in precipitation in equivalence zone. And then, after we increase the amount of antigen, there will be nobody to bind with antigen. So the antigen excess zone, that means antigen is the antigen quantity is more than antibody. There are not enough antibody to bind with antigens. So the precipitation will be decreased until the last tubes there will be no precipitate in that case. By plotting the amount of precipitate against increasing antigen concentration yield a precipitation curve. We can make precipitation curve and to measure the precipitation curve in that case. The precipitation curve when increasing concentration of antigen added to, as we mentioned in the previous slide, to a series of tubes contain a constant concentration of antibody, variable amounts of precipitate form. Precipitin curve can be obtained by plotting the amount of precipitate against the amount of antigen added, and we can measure the precipitate in that case. Precipitation reactions in gel. Some examples: radial immune diffusion, immunoelectrophoresis, Western blot, immunoblot. You are going to take details about Mancini and Octoloni precipitation and reaction in gel in the laboratory lessons. So we just mentioned some examples, and the details about these reactions will be discussed during the practical lesson. Immunoassays, direct binding immunoassays like RIA, radioimmunoassay, which is no longer being used because it's very difficult and radioactive substance needed in that test. Solid phase immunoassay, ELISA practical, it's used in hospital lab, in laboratory for detection of infection or for many practical use. Immunofluorescence also as type of immunoassay. Flow cytometry is also. All these will be discussed during the practical lesson for you. Thank you very much.